Hi everyone, my name is Jalal Jeber. I am a faculty member in University of Baghdad College of Science Department of Chemistry, Iraq, Baghdad. Today I am going to present my paper which has the following title: On-site detection of saliva alcohol as a function of the blood alcohol concentration using colorimetric biosensor based on deposited chromium oxide nanoparticles on a filter paper. This paper is participated and published as a part of the 8th of the International Conference of Biotechnology, Environment and Engineering Science, which is held in Stockholm, Sweden, 18th of October 2020. So the outline of the present study is covered the following points. First, the main objects of the current presentation, and then we're gonna move to brief introduction, and we'll explain the experimental section and the result and discussion, and end up with a conclusion. So the main objectives of the present study is the following 3 points, which are: first, the development and validation of a new method for determination or detection of a blood alcohol concentration. The second point is the detection of a blood alcohol concentration in saliva. As we know, in order to determine the concentration of blood alcohol concentration in blood, we have to take a sample of blood, and this process is very long and time consuming. Therefore, detection of blood alcohol concentration from the saliva will be a powerful tool for determining this type of analyte. And then the following technique is very simple and rapid and low-cost method in comparison with the previous or reported method in the literature survey. So the introduction will be about some historical information about the conception of the alcohol. Therefore, this consumption will lead to the risk factor for morbidity and mortality which related to the both unintentional and intentional injuries. Globally, and according to the WHO, World Health Organization, 16% of death and 13% of disabilities from injuries were estimated in 2000 to be attributed to the alcohol. Alcohol can coordinately affect many individual psychomotor skills such as brain, hand, food, and eye. The effects of the alcohol on human body are several, such as visual focus, long reaction time, and delay in the judgment, leading to the injuries from causes like falls and motor vehicles accidents. Therefore, all these introduction will make a reasonable for conducting the current study, which are the BAC responsible for many of accidents such as drawing, falls, and so on. And the second point, the BAC can explain the occurrence, lots of types of injuries, and end up with the development can effectively be used in many purposes such as forensics and the medicine. The experimental section in the present paper will include the following points. The first and 2 points is about how you can synthesize and characterization of nanoparticles. These particles we're gonna use on the surface of the filter paper that is used as a biosensor. And then we're gonna load a few amount of the chromium oxide nanoparticles after synthesize it and characterize it by using different technologies such as AFM, which is the atomic force microscope. These nanoparticles will be deposited on the filter paper. And then preparing the samples, which means the people of the kind of people which the current study is covered. And then the development of the general procedure, which is the procedure will be followed by the author. and then validation of the proposed method, and end up with the application of the proposed method in real sample. So the first part in experimental section includes the following details: synthesis and characterization of the chromium oxide nanoparticles. As you can see, there are 4 pictures in this figure: A, B, C, and D. The first, A, is the empty cup without anything, and the B figure, it is the filter paper that we have used in order to prepare the biosensor. And you can see the chromium oxide nanoparticles have a colorless color, and we took a few amount of it after prepared it and put it on the surface of the filter paper. And the figure C and B, as you can see, it is the picture of atomic force microscope, how we can characterize the nanoparticles, and the nanoparticle size that we synthesize is about have the following range from 70 to the 80 nanoparticle as a diamond. Also, the experimental section include the following parts, which is the loading of chromium oxide nanoparticles into the filter paper. The first and the second stages, A and B, is considered as the initial stages. And then, as you can to the figure C, to the yellow color, which is the reaction between the chromium oxide nanoparticles and sulfuric acid, which were used as acidic medium. And after adding the sulfuric acid to the filter paper, the color will change from the colorless to the yellow color as a result of the reaction between the chromium oxide nanoparticles and the sulfuric acid. And you can see and consider the figure D is the final shape of the biosensor that we have prepared. Also, the samples in this experiment covered the following number. The total number of the participants in the current study is the 50, with the age range from 25 to 45 years, which divided into the 2 groups, Group 1 and Group 2. The Group 1 consisted of 20 participants. 10 out of them are from male and the other from the female, while the Group 2 consisted of 30 participants. 20 them are from male and the other from the female. Result and discussion. For the first group, 19 out of 20 of the Group 1 samples, which represent 95%, have demonstrated a positive result, with variable intensity of green bluish color, while the remaining 50% show a negative result. While Group 2, 24 out of 30 samples, which represent the 80%, have shown a positive result, and the remaining 10% show a negative result. Here we need to clarify what does we mean by positive and negative results. If you can see to the figure A, a green color means we have a positive result, while the yellow color means we have a negative result. Therefore, any result that have a green color means we have a positive result, and so on for the yellow color. Also, in the result and discussion for Group 1, as you can see, we have different types of beverage that's covered in the current study. And each type of it, there are a total number of samples. And also there is a number of positive and number of negative. As you can see to the section in this table, which is the found in serum of the positive samples. each samples that tested positive at the same times we have compared the positive result with the result that obtained from the reference method, which is the gas chromatography method. Therefore, any positive result will immediately characterize or determine the blood alcohol concentration in the serum. Therefore, as you can see to the tables, we have 20 participants or samples as a total number. As you can see, there is an opposite side, there is a concentration that is found in the serum of that sample. While group number 2, we have different numbers. Sum of numbers or the total numbers of participants is 13. The number of negative samples is 8. And these samples are determined or obtained as a negative result, which means the biosensor color is still yellow, while the other species or samples, the yellow color of the biosensor are converted from the yellow to the green, which means we have obtained a negative numbers. As a conclusion, a colorimetric saliva-alcohol biosensor has been described in the present study. The disk is a rapid, practical, and feasible method for the detection of a blood alcohol concentration of 0.0% or higher throughout the saliva for all types of the sample, even though for those with unconscious stasis. The third point is the above description procedure exhibits the capability of the method for the detection of alcohol in saliva on site without any necessary pre-treatment. The final point is the disk is simple, low cost, and rapid, and it must be used in forensics or in the medicine purposes as a powerful tool for blood alcohol detection. And the final parts in this presentation is some of useful references that we use in our papers. And thank you for listening.